notes on protein synthesis. This is actually section 17. All right, remember I said that protein synthesis is transcription and translation. So we're going to do transcription first, where we're going from DNA to mRNA. mRNA is messenger RNA, is transcribed from a template strand of a gene, which is just a section of DNA. RNA polymerase is going to pry the two strands of DNA apart, similar to replication, and joins together RNA nucleotides complementary to the DNA to that. This is actually RNA polymerase 2 because eukaryotic cells have more than one RNA polymerase. It's only going to happen in the 5' to 3' direction, which is important in how it reads the amino acid sequence. If it didn't read 5' to 3', it would be like reading a sentence backwards. We don't need an RNA primer because at this point we're not inserting DNA nucleotides. It's always going to be RNA. So how do we initiate this process? In each of transcription and translation, we're going to talk about initiation, elongation, and termination. So how do we start it? How does it happen? And how do we end it? The promoter is a sequence of DNA where RNA polymerase will attach and begin transcription. The start point is where the actual RNA synthesis will begin. In eukaryotes, proteins called transcription factors are going to mediate the binding of the RNA polymerase and the initiation. So together, the transcription factors plus the promoter are called the transcription initiation complex. The promoter of a eukaryotic cell is typically a ta-ta box, which is just an area of lots of repeated T's and A's. Transcription factors recognize that area, and that's where they're going to bind to start the process. So here's a big picture of the ta-ta box. So just repeated T's and A's. This is going to tell the transcription factors bind here. And then at some point right upstream of that is going to be the actual start point of where we're going to start transcribing that gene. So here we've got RNA polymerase binding. We've got our transcription factors. We're ready to go. We're going to break the DNA apart. We're going to go 5' to 3', and we're going to start building our RNA. RNA polymerase will continue to unwind the DNA and add the RNA nucleotides to the growing mRNA strand. The new mRNA is going to peel away from the DNA, and DNA will go back to its double helix form. A single gene can be transcribed simultaneously by several RNA polymerase molecules, and that means that you can make a large amount of protein. You can make lots in a short amount of time. So here's a picture of this. So here's where we're going, 5' to 3'. We've unwound the DNA, that's blue. The RNA here is in red. Okay. We've got our RNA polymerase, that's this big blob here. And you can't see the transcription factors anymore in this picture. But we're just going to keep moving forward with this process and continue to unwind the DNA here. And we're going to pair up, and remember it's complementary. The only difference here is that remember there's no T in RNA, so you're using it. How do we end this? Well, it differs a little bit from bacteria and eukaryotes. In bacteria, they're going to go through a sequence that is called a terminator sequence. It's just a sequence of DNA that is recognized as being, oh, this is the end. In eukaryotes, RNA polymerase 2 is going to transcribe the DNA sequence called a polyadenylation signal sequence, which codes for a polyadenylation signal. So it's A's with a U in there. Several nucleotides later, the RNA transcript is going to be cut free, and that's going to be our pre-mRNA. Eukaryotes are then going to create a pre-mRNA, which must then undergo modifications. We call that RNA processing, where both ends of the pre-mRNA are altered and certain sections are cut out, and the remaining portions are spliced together. The 5' end of the pre-mRNA is going to receive a 5' cap, and the 3' is going to receive a poly A tail. That's just a string of a lot of A's. Both of these seem to, is what scientists think, facilitate the export of the mature mRNA, so being able to be recognized and leave the nucleus. It also protects the mRNA from degradation by enzymes because it's protecting that gene. It's like putting a cap on both ends to protect that precious gene on the inside. It helps the ribosomes attach to the 5' end of the mRNA. So here's what we have. This is the 5' cap. This is your poly A tail. This is actually your gene right here. Okay, this is your sequence that you've made. This is that termination sequence, the polyadenylation signal, that it has to go through. Notice that both of these here are called UTRs. Those mean untranslated regions. So those are actually sequences of DNA, A's, T's, G's, and C's, but they're not actually transcribed and translated. Untranslated regions. RNA splicing also happens that removes portions of the RNA. DNA molecule is about 27 nucleotides for a gene, but it's only going to take about 1,200 of them to code for a protein on average. Eukaryotic genes have long non-coding regions that are cut out. Those non-coding regions are called introns. And exons are what are eventually actually going to be expressed or translated later on. Introns are cut out, exons are joined together, and that's a little hard to remember. Introns versus exons that you do know that. Exons joined together, introns cut out. Let's do that. So remember we said we have the cap, we have the tail. The introns are the uncoded regions, and so those are going to be taken out, and then only the exons are going to be put together. So this ends up being actually a little shorter than what the entire sequence was from beginning to end. And how does that kind of happen? We use SNRNPs and other proteins. These are just proteins that help. They form something called a spliceosome, and that will create this little tail here and basically slice it out. And there's a cut out introns. And then exons are put back together. All right, so that is transcription. We've got a mature mRNA. It's leaving the nucleus. Now we have to translate it. This is where the cell is going to read the message and create a polypeptide. The message is a series of codons on the mRNA. We talked about that. And the tRNA is going to transfer the amino acids from the cytoplasmic pool of amino acids to the growing polypeptide chain. So you just got all these amino acids floating around in your cells. And they're going to bring those amino acids to the ribosome and attach them to the ones that are already there. Let's see this. RNA is going to arrive at the ribosome carrying an amino acid. tRNA or transfer RNA. It transfers amino acids to the ribosome. The other end of the tRNA is called an anticodon. It's going to be 3 bases as well, and it's going to pair up with a codon that is on the mRNA. The tRNA is also transcribed from a DNA template. There's a whole gene that's all about making tRNA, and it's made in the nucleus of eukaryotic cells. Each tRNA is used repeatedly, so when it's done, it goes and it finds another amino acid. This is tRNA. Anticodon is 3 bases here. It's going to match up with the codon on the mRNA. There's lots of ways that books and animations and stuff show what tRNA looks like. Some show it here with all of the actual nucleotide bases. Some of them are going to show it more theoretically with just little notches to indicate that. And then some of them are just all together going to show it as a big blob like this. This probably makes it a little simpler in understanding when you actually see the process, rather than having to see all of this and get all these loops and holes and letters and get it confused with other parts of the process. So here we go. Here are your amino acids. They're picked up on the tRNA. The tRNA is going to come to the ribosome. It's going to match up with the codon on the mRNA. This is remove the anticodon. They're going to pair up, and it's carrying an amino acid on the end. When it's done, it's going to leave behind the amino acid, and we're going to continue to grow that strand as we are moving along the mRNA and translating it. The correct matching of the tRNA and the amino acid is carried out by amino acid tRNA synthetases. It's just enzymes that happen. Only about 45 tRNAs. So we have 60 amino acids, 45 tRNAs. Those tRNAs have to be a little bit more flexible and pick up more than one possible codon, be able to pair up with more than one possible codon. That's why we have a wobble position. The third nucleotide base of the codon, and the codons on the mRNA, is going to correspond to the base of the tRNA, and those are flexible in their base pairing rules. So typically A pairs with U in RNA, and G pairs with C, and this is going to be a little bit more flexible in that position. Let me show you a picture of that here in just a second. For example, the tRNA with anticodon GAG, right here, can base pair with either mRNA codon CUC or CUU. So CUC is technically what that should base pair normal pairing, but that wobble position allows it to be flexible in its base pairing, so it can actually pair with a U and it still be okay. Because remember, there's only 45 of these little suckers, and there are 60 codons for the 20 amino acids. Ribosomes are actually composed of two subunits. One is large and one is small. Each subunit consists of proteins and one or more ribosomal RNA. So we're adding another idea of another RNA. In eukaryotes, the subunits are made in the nucleus. The large and the small subunits only join to form a functional ribosome when they attach to the mRNA. The ribosome also has three binding sites: the P site, the A site, and the E site. The P site is where this is like in the middle, and this is what actually holds the tRNA that's going to have that growing amino polypeptide chain, amino acid chain. The A site is going to carry the next, or it's going to accept the next tRNA, and the E site is where it's going to leave or exit. So it's like accept, which is not probably the right word for accept, but it fits with the A. Exit, and I don't know, producing, because it's producing the chain here. So tRNA first comes into the A site, moves over to the P site. This is where you're going to have all the amino acids, and then it's going to move to the exit site, and then it's going to go away, and another one's going to be coming in, and it's going to keep progressing in that direction. Small ribosomal unit binds to both mRNA and a specific initiator tRNA, which carries the amino acid methionine. Remember, that's always the beginning of the gene. Then the large subunit is going to bind. The initiator tRNA binds to the 5' cap and moves along until it reaches the start codon, AUG. That's going to establish the reading frame for the mRNA. Remember, that's very important, that every 3 is read correctly, or your sentence is not going to make any sense. Excuse me. So here it's showing you that this is your codon and your anticodon. Got your amino acid here, and this is the start codon. It will start when it hits that AUG sequence. The initiator tRNA site sits at the P site on the ribosome. The polypeptide is always synthesized in one direction from the N-terminus toward the final amino acid at the carboxyl end, or the C-terminus. So in any amino acid, this is going to be the N-terminus, and then this is going to be the C-terminus, the carboxyl acid. So all of your amino acids are going to be in that direction. Because notice, these amino acids are just little shapes. But we all know, based on our chemistry knowledge, that amino acids are actually made out of a carbon, a hydrogen, an R group, which is different in all of them, an amino group, and a carboxyl group. And so this is going to be N-terminus, C-terminus. All the amino acids are going to be in that order. N on one, C on the other, starting with N and ending with C. Amino acids are added one by one to the previous amino acid at the C-terminus of the growing polypeptide chain. So you're going to add the amino acids there on this end. Each one will form a bond here. The amino acids are joined together by peptide bonds. That's going to join them. The ribosome and the mRNA are going to move relative to each other unidirectionally, 5' to 3' codon by codon. Very choreographed. So amino acid or tRNA in, carrying amino acid, form a peptide bond, add to the growing chain, tRNA goes out, and it's going to go find another amino acid. And it'll just keep going in this direction. So here we go. You can see the tRNA coming in with the amino acid. Peptide bond forms. This one moves over, this one moves over, and then that one's now exiting. You can kind of see that play by play. We'll still watch some animations to help you with that. Elongation continues until a stop codon on the mRNA is reached at the A site. Because remember, the A is the acceptor, and then the P site's where it progresses, and then the exiting is the E site. It releases the factor, causes the addition of a water molecule to the polypeptide chain instead of amino acid. This hydrolysis reaction will break the bond between the completed polypeptide and the tRNA at the P site, releasing the polypeptide through the exit tunnel of the ribosomal's large subunit. So here's our exit tunnel right here. It's going to break that off and leave polypeptide. So there's just an amino acid chain. And then the ribosomal subunits will break apart and release that mRNA, and mRNA will be degraded. Multiple ribosomes will translate an mRNA at the same time. Again, just like before with transcription, that allows a cell to make many copies of the amino acid sequence very quickly. So this is a microscopic view of this happening. Each of these little suckers is ribosomes. And this, it looks like string going through them, is your mRNA. So it will, lots of these are going at the same time. So we can get lots and lots and lots of proteins made all at once. And then we have protein folding. So on page 82 is going to provide you details that we've talked about before about levels of protein structure. Remember, your primary is your sequence of amino acids. Think about this as your string of pearls, and each pearl is an amino acid, and the string of them together is your primary structure. Your secondary structure is going to be when you coil and fold that polypeptide. There are alpha helixes and beta helixes, which are examples of ways that they can be folded. The tertiary is going to be, once it's folded, the overall shape resulting from the interactions of all the side chains. Remember, those R groups are really important now. And how they interact is going to produce your tertiary structure. Your quaternary structure is going to be a result of, here's one sequence, here's another, and how do those two sequences interact together. For example, hemoglobin has two amino acid chains, or polypeptides. So primary, secondary, tertiary, quaternary. All proteins have these. This one depends on the protein. There may or may not be a quaternary structure. Post-translational modification. This may or may not be required. Certain amino acids may be chemically modified by attachment of sugars, lipids, or phosphate groups. Enzymes may remove one or more amino acids from the end of the polypeptide chain, or the polypeptide chain may be cleaved into multiple pieces or come together. That would be that quaternary structure. Targeting polypeptides to specific locations. Remember this from unit 2 or 3, 2, I think, where we talked about all the different parts of the cell and what they do. Where the free ribosomes are mostly going to synthesize proteins that are going to stay inside the cell. The bound or attached ribosomes, those are the ones that are on the ER, are going to make proteins that are going to be secreted from the cell. Why is that? Well, that is because on that ER, when those ribosomes make it on the ER, they can form a vesicle, and the vesicle can fuse with the cell membrane and release the contents of that cell out. They can switch roles, however, but that's typically how it goes. SRP, signal recognition particle, is an escort that brings the ribosome to a receptor protein built into the ER membrane. So it brings the ribosome to a receptor protein that's on the ER membrane. So here we go. SRP is right here. It's going to bring those ribosomes there, on there. They're going to synthesize proteins, and then they're going to excrete them out of the cell. Typically. We talked about a lot of different types of RNA, so this just kind of summarizes some of those RNAs so you don't get them confused. mRNA is involved in transcription. Transfer RNA is involved in translation. Ribosomal RNA is part of the ribosome structure. We talked a little bit about the primary transcript. Remember, like the pre-mRNA versus the mature mRNA. And then we also talked about snRNA. These are structural and catalytic roles in spliceosomes. So we're removing the introns and putting together the exons. So I want you to try to work backwards. Using figure 17.5 in your book, I want you to identify the 5' to 3' sequence of a nucleotide in the DNA template strand for the mRNA coding for the polypeptide sequence P, pro, L, lyse. So you have to take these, use that codon chart, okay, that we had in here, and I want you to find what the mRNA would be, and then from there, what would be your original DNA template strand. So if you can do it.